is a native of Bolivia and then received his undergrad degree in Guadalajara in Mexico. He then moved to the U.S. to the University of Arizona in Tucson, where he received his PhD in analytical chemistry and then stayed on for a postdoc with the great bioinorganic chemist Ann Walker. He then began his independent career at Oklahoma State University and followed that with a move to the University of Kansas, and then found his way back to the South. And he's been in the Department of Chemistry here since 2018. And with that, it's my pleasure to turn it over to Dr. Rivera.
Thank you, David. Can you hear me in the back? Yes? Excellent. Thank you for the invitation. I was looking forward to giving this talk here in biological sciences. Although my home is the chemistry department, I think I have a lot in common with biochemistry and biology. So looking forward to this one. And what I want to tell you today is a story that started about 15 years ago. It was just a project on understanding structure-function relationships in an iron storage protein. And then over the years, it turned into what is now a drug discovery project. And so I'm going to take you through how that happened and where we are today. So it's sort of a project progress report. So why is this not advancing? It was something we had to do earlier? I can't remember. Sorry. Okay, there we go. So, thanks, Dave. So, basically, what I want to tell you today is about how one can use iron homeostasis as a potential target for developing antibacterials or antibiotics. And although antibiotics were our initial thought, as it turns out, our molecules or our target seems to be more important to addressing biofilms. And so biofilms are bacterial communities which are embedded in extracellular polymers, which is called the EPS, right? And so one thing that's important to understand in this context is that bacteria in biofilms are phenotypically and physiologically different from bacteria in free-living bacteria, also known as planktonic cells. So these biofilms enable bacteria to colonize and persist in the environment. As a matter of fact, the biofilm, although relatively recent in terms of laboratory experiments, biofilms are probably the preferred way in which bacteria live. They will form biofilms which will allow them to survive very challenging situations in vivo almost anywhere. And so planktonic cells, on the other hand, seem to be more like a laboratory situation where we can grow cells in rich media. And so normally cells are not going to find LB in their environment or anything that rich. So they will turn to form biofilms. And so these biofilms can develop at a solid liquid interface, like they will attach to a surface and then they will form the biofilm on a surface. We've seen this hiking around, in a stream. You can see when it's slimy, that's probably a biofilm in a rock. Also, they will grow at their liquid interfaces, right? And when they grow, they attain all these properties. This is a nice high-resolution picture of a biofilm, just to remind you of it's a three-dimensional architecture, okay? And so important things about biofilms, some other important things. So these cells that are embedded in biofilms, they can be up to 1,000-fold more resistant to antibiotics than planktonic cells. And that is really an astounding difference, because a lot of, for example, sensitivity testing that's done for antibiotics is done in planktonic culture. And when one does the experiment, I'll show you some examples with the right or with the properly grown biofilm, then it turns out that bacteria that are susceptible to antibiotics in planktonic culture are no longer susceptible in the biofilm. And but this lack of susceptibility is a term that we're going to use, tolerant. So they're tolerant to antibiotics, and tolerance is different from resistance. Okay, tolerance is a physiological condition not involving mutation, but the physiology of the cell in the biofilm. On top of that, we have that tolerance, right, can be mixed, or if bacteria that carry a resistant impairing mutation, that is an antibiotic resistant strain, can also form biofilms. And so the situation is compounded when these multi-drug resistant bacteria can form biofilms. We have been studying Pseudomonas aeruginosa. It's a gram-negative, it's an opportunistic pathogen, and it has a large propensity to form biofilms. As a matter of fact, Pseudomonas aeruginosa biofilms are probably some of the best understood biofilm-forming bacteria in the literature. So also another important reason why Pseudomonas is worth studying is there's a recent report that's currently highly cited by the WHO, right, where Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacteria, they are gram-negative organisms for which antibiotics are desperately needed. And we're not yet talking about antibiotics. So it's multi-drug resistance. And so gram-negatives have been notoriously difficult to develop antibiotics against. And one of the reasons that often cited is that gram-negatives are more difficult to permeate because of the two membranes. Okay, but that's not just as simple as that. You can see that, for example, E. coli will have another membrane envelope that is easier to permeate, so to speak, than Pseudomonas. So Pseudomonas is notoriously difficult to permeate. Medically speaking, in terms of biofilm infections, biofilms in the cystic fibrosis lung are devastating. There have been enormous advances in cystic fibrosis and how to treat cystic fibrosis patients. But one of the remaining challenges is still bacterial infection. So over the years, the lungs of cystic fibrosis patients get colonized with Pseudomonas aeruginosa, and many times that is the reason for death, not the cystic fibrosis necessarily. And these are biofilms in the cystic fibrosis lung. Ventilator-associated pneumonia, indwelling medical devices, that's another difficult situation where sometimes if a biofilm forms in an indwelling device, the only way to get rid of the biofilm, is to just remove it, okay? And also in wounds, burn, pressure ulcers, diabetic ulcers. You have probably heard about diabetic patients, for example. If the pressure ulcer or the diabetic foot ulcer, as is known, gets infected and a biofilm is developed, those cells become really impermeable or untreatable with current antibiotic technology. And so that leads sometimes to amputations and situations of that sort. So there's really a very important need to develop molecules or treatments for biofilm bacteria. And so we have been using iron homeostasis as a potential target, and this started many years ago, as I said. And but let me explain why iron homeostasis may be a reasonable target. So it turns out that iron is essential for us and, of course, for bacteria. And so our immune system, using molecules like transferrin, ferritin, lipocalins, and hemoproteins, really sequesters the iron at very, very low levels. cells, such that bacteria need to, in the absence of iron, it cannot really colonize or set an infection. Of course, among the many types of bacteria, pathogens have evolved biochemical ways to overcome that immunity, which is carried out through for example, the release of siderophores. Siderophores are chelating agents with very high affinity for the iron 3 ion, right? 10 to the 40, sometimes larger. So they can really, once they release, they can sequester iron 3, bring it into the cell. Porins can sequester and internalize iron 2 when that's available, and that will depend on oxygen concentrations. And heme is sequestered by hemophores, and heme is a rich source of iron, except that the heme iron now has to be released. So this is something that we found in the lab a few years ago. Biofilm cells require twice as much iron as planktonic cells. So really the requirement for iron becomes more stringent in the biofilm lifestyle. And so we asked this question several years ago: bacterial iron homeostasis, is it a potential target for developing antibiofilm? And the initial question was really antibiotics, okay? And so this is just a reminder of a schematic of iron homeostasis in bacteria. And so you have, this is a gram-negative, you see the two membranes. This is a situation, for example, where hemophores can be secreted, sequester the heme from hemoglobin, bring the heme in, then an enzyme like heme oxygenase can break down the microcycle and then release iron 2. This would be siderophores, and these siderophores are secreted. They can sequester the iron 3, bring it in. It's a reduction in the cytosol, and then basically going to this cytosolic iron pool. This is also known as the free iron pool. And that is different from the total iron in the cell. The total iron in the cell will consist of the cytosolic free iron and also all iron that's incorporated into heme-utilizing proteins, many kinds. The most common are heme, iron sulfur clusters, mononuclear, dinuclear iron centers, many, many ways in which iron participates in all kinds of biological processes within the cell. And then something unique to iron has been the invention of iron storage proteins. So normally other metal ions, transition metal ions, and important metal ions in cells, they haven't been given the honor by nature of inventing a molecule just to store that metal ion. The only one is iron. And that has to do with the chemistry of iron, where iron is essential. Iron in the +2 oxidation state is soluble at pH 7, but highly reactive with oxygen, forming oxidative stress, right? And iron 3 is highly insoluble, extremely insoluble. So those two properties make iron very difficult to handle by the cell, but because things evolved initially in life, apparently, in the absence of oxygen, iron had already been used for many, many types of proteins and enzymes, and so this invention probably comes a little bit later. And so this cytosolic iron, it was thought to be stored somehow in here, and then before we started in the field, it was not known how the iron, once needed, would be released. And so that's a question that we started working on when we started here. And so this cytosolic iron, via the master iron regulator FIR, it's going to repress or de-repress the genes for iron acquisition, okay? And so a lot depends on a healthy balance or a healthy concentration of iron 2 in the cell, free iron. So this bacterioferritin is a very large molecule, about 500,000 Daltons. It assembles from 24 identical subunits and 12 hemes. It is a full sphere. in this sphere, I'm showing you a transverse view here. In this sphere, this molecule theoretically can store up to 3,000 or 4,000 iron ions. Okay, so the hemes, what's unique? So you've heard of ferritins. Bacterioferritin is unique in that it contains hemes, 12 hemes, and very little amino acid sequence similarity with ferritins. So this is a subunit in bacterioferritin, a 4-helix bundle with a perpendicular helix here, and then two of these bind one heme molecule, okay? And in the middle of each one of these subunits is what we call a peroxidase center. And the peroxidase center ligands are here. And the way that works, iron 2 comes, gets into that peroxidase center, with oxygen or peroxide is oxidized to iron 3, a diferric center, and then the iron is gated inside the cavity where it forms an iron mineral, okay? In order to release the iron, we found out that there is the need for a protein-protein complex where the large bacterioferritin binds to a very small 8 kilodalton ferredoxin. So a ferredoxin is a small molecule, a small protein that contains a 2 iron 2 sulfur cluster. I'm showing you the 2 iron 2 sulfur cluster in orange and yellow here, right? And it binds in between 2 subunits, and we calculated that these 2 amino acids in purple would be the best way for the electron to travel across this insulating barrier of the protein to the heme and through the propionates into the iron, so that the iron 3 can be reduced, right? And so because there are 12 hemes, we found that actually the structure shows 12 BFDs can bind, 1 BFD binding per heme, right? And so then, in summary, the function of bacterioferritin is to take iron 2 and with using oxygen or peroxide, oxidize it and store that iron. So that in itself has antioxidant properties, right? And then when needed, BFD can bind in between a subunit, as we're showing here, and the electron travels through the heme into the iron core, which is ferric, reduces the iron 3, and the iron 2 can be moved out, right? And then incorporated in metabolism. So this complex, once we solved this structure and we saw the importance of this, and at that time we had all the information mainly in vitro, brought the question, can we target this protein-protein complex for the development of new antibiotics? And what would be the advantages of doing that? For example, other efforts out there in the literature by other groups have been centered on targeting iron acquisition. Since iron is essential, and then the immune system is already helping in reducing that concentration of available iron, can one interfere with that? And the answer is possibly yes. But what are the disadvantages? The disadvantages are that the ways by which bacteria acquire iron are multiple. So if one interferes with one, then another one can be upregulated. And so it becomes very difficult because one would have to interfere with them all. So we thought that inside the cytosol, is this going to be a good target for us to dysregulate iron homeostasis? And before this experiment, nobody knew what would happen. There had been experiments where people added chelating agents, and you can really sequester the iron. But if those chelating agents also permeate the cell, right, then you're doing things that are not easy to do in the case of an antibiotic, because you don't want to send chelating molecules that also chelate the iron and other metals in the patient. So we thought that what would happen here, we were wondering. And so we needed to, in order to target that, we needed to understand it a little bit better. So the first thing we needed to understand is if we're going to develop molecules to stop these two proteins from interacting, what are we against? Nanomolar affinity, micromolar affinity, what is the affinity? So we measure the affinity of these two proteins interacting using surface plasmon resonance. Here's the schematic where this molecule is symmetrical. So we just immobilize the bacterioferritin on the chip. Then you can flow the BFD molecules through the cell. And then as you increase the concentration of BFD, you get a nice response. Out of these response, whenever you're in the steady state, then you can plot or you can obtain a binding curve. And so to eliminate some of the details, the important thing is that this curve is fitted with a model where all the 12 BFD binding sites are independent. Okay, there's no cooperativity or any situation like that. They are completely independent. All bind with a KD of 3 micromolar. So that's the other thing. It's not a tremendously high binding affinity, so it should be reasonable to interfere with. Okay, and why not so high? Because these are electron transfer molecules, so they have to come together. The electron transfers, and then the molecules separate for the next cycle to occur. In most cases. So looking into the details of the iron of the interface, what I'm showing you here is the bacterioferritin in surface rendering in green and in gray, okay? And those residues that are pertinent in BFD, the partner protein, are in cyan sticks, and the iron sulfur cluster in BFD is in gold and orange and yellow here. And so what we noticed was that this leucine 5 and this tyrosine 2 bind in this cleft. And this cleft seemed to be very interesting from the point of view that that's probably what we could target for developing molecules that stop these two proteins from interacting. But that was just a hunch. So what we needed to do is find out what are the hot spots of the interaction. In any protein-protein interaction, you have a number of residues, which is not all of the residues at the interface, which dominate the energy of the interaction. The rest contribute a lot less. And so we did sort of an alanine scanning mutagenesis experiment, and so I'll just show you some of the results here. So we can measure the KD for the interaction of the wild type at 3 micromolar. And so we looked at this cleft here, which is formed by the side chain of glutamate 81 and the side chain of leucine 68. Okay, and so we mutated one at a time, and so you can see almost 100-fold increase in the KD. If you do the double mutation, these two residues to alanine, then the interaction becomes no longer measurable. So these are hotspot residues, and so we decided to pursue these. And so what happens if there is no interaction? In vitro, we did an assay where the bacterioferritin is loaded with anywhere between 500 and 800 iron atoms in the interior cavity. Then we reconstituted the bacterioferritin with BFD, okay? And then we add NADPH. And if you do this in a glove box, you don't have to worry about oxygen. So you can see that the iron comes out. Basically, all the iron in the cavity is released as iron 2. So if you work with any of the bacterioferritin mutants, the single mutants, you can see that the rate at which the iron is mobilized has changed. Obviously, the interaction has changed. And then if you work with the double mutant, the leucine 68, glutamate 81, both to alanine mutant, basically the curve you get is superimposable on the one you get if there is no BFD. So basically no interaction, no iron mobilization. So these experiments demonstrate that's important. But these experiments were in vitro, and so now the question is this the only mechanism by which iron is mobilized from bacterioferritin in Pseudomonas aeruginosa? And to answer that question, we did a few things. First is we use gel electrophoresis, but this is the staining here is important to describe. So what we did is, since we can produce the recombinant bacterioferritin, we can then separate it in a native gel, and then we will stain not with Comasi, but with Ferrin S. So Ferrin S is a stain that's going to allow you to detect iron that's concentrated in certain places. And so since iron is concentrated in the bacterioferritin, then the blue stain is the blue complex of Ferrin that develops because of the concentrated iron there. So what we can do then is culture, and these are planktonic cultures of Pseudomonas, which are harvested at different time points. Those cells are harvested, are centrifuged, they're lysed, and then the clarified supernatant is loaded on the gel. And then you stain with Ferrin, and you can see that there's accumulation of iron in the late stationary phase. And then as the iron in the culture media begins to disappear, then actually the iron in the bacterioferritin gets mobilized, and then it's used by the bacteria. So we have then the same experiment with a knockout mutant where the BFD gene is no longer there. And what we find is that the iron accumulates irreversibly in the cell. So this shows then that no BFD, no BFRB, no BFD interaction, the iron stays there. So we also, taking advantage of what we had learned in vitro, we did a double mutation in the chromosome where the BFRB gene has been replaced by this allele, where the two mutations here, so we call that BFRB star for short, right? And you can see that now the two proteins are present in the cell, but because of the mutation on BFRB, there shouldn't be any interaction. And indeed, the iron is irreversibly trapped inside the bacterioferritin. And so then we went to do some of these measurements. And so we measured total intracellular iron, okay? And so we measured at 12 hours and at 18 hours, okay? And what we find is that we have the wild type and the two mutants. And there's maybe a little bit more iron in the mutants than in the wild type, right? Same situation at 18 hours. And this is the classical experiment that most people do. So this would have not told us very much. What really was significant was to go and use EPR. And actually, David, you helped with these experiments with the EPR. I don't know if you remember. Annabelle and Hui Li, with David's help, measure by EPR the intracellular iron. And so at 12 hours, the intracellular iron in the mutants is lower, significantly lower than in the wild type. And that situation becomes even worse at 18 hours. So what we are observing at this point is that the intracellular iron is telling us the story. Why not the total iron? Because this is telling us iron starvation, but the total iron is also considering the unusable iron trapped in the bacterioferritin, right? So this measurement is not all that revealing, but this is. And as we would expect, if we have an iron starvation response, then pyoverdin is secreted by the mutants in enormous proportions. And this is a non-ribosolvable synthesis, very energetically costly. So to have these large differences in pyoverdin secretion indicates that the cell is really, really hungry for iron, okay? So then we have irreversible trapping of iron and iron starvation as a consequence. So with that, we propose to revise a little bit the conceptualization of bacterioferritins. So normally, bacterioferritins and ferritins have been referred to as cages, where iron is just put in, right? And then somehow, when iron is low, this iron can come out. So as a matter of fact, we propose a different situation. What we propose here is a dynamic equilibrium between the iron 2 in the cytosol and the iron 3 in bacterioferritin. And this dynamic equilibrium, of course, depends on these two situations: oxygen or peroxide to oxidize and store the iron, BFD to release the iron. And this equilibrium should be able to buffer the iron, the free iron, at a concentration that matches the KD of the master iron regulator, which needs iron, right? It is the ferrated complex that actually tells the cell, we're happy with the iron, we don't need to activate all the genes to bring iron, okay? But if we mess up with this equilibrium, then what happens is this becomes a reaction that goes mostly in this direction, where the iron is stored irreversibly here. This concentration drops below what's needed here. It's no longer a regulatory concentration. So this master regulator is sensing iron constantly, despite having quite a bit of iron in the cell. So this buffering capacity is eliminated like any other capacity, buffer capacity, if you mess up with the equilibrium in the buffer, right? This just happens to be an iron buffer. So now that we know that those are the deleterious consequences to the cell, we wanted to see what would happen in biofilms. And other work in the lab had already told us that biofilms require twice as much iron as planktonic cells, so we wanted to test this. And so we developed a way in the lab to study these biofilms. And so what Annabelle did is take, for example, a petri dish like this, and we will grow the cells starting with a planktonic culture without shaking. And I said earlier that biofilms develop nicely also at air-liquid interfaces. So what happened is a biofilm develops at this interface. And so you can see it here. You can see the biofilm. And these cells have secreted a tremendous amount of pyoverdin and pyocyanine. That's why they're so green. And so you can see this. And so now the question became, how do we harvest this? And what advantages it might have? So the solution was to take a cover slip and then go on down and gently touch the surface where the biofilm is. And the glass really attracts this biofilm, and the biofilm just gets attached to that glass. We can take this glass and we can do many things, right? So one of the things that we can do is we can now take the biofilm and now make it float on top of on a solution containing antimicrobials. So we can test antimicrobial sensitivity. We can also mount it on a glass slide, and we can do confocal microscopy that we have been doing quite a bit. Or we can take this glass and then we can just disperse the biofilm by sonication or other ways of dispersing. So this is quite enabling to work with biofilms. Relatively simple growth, and then one has to be careful about this harvesting step, but then when things go well, normally the experiments work. So the initial work was done by Annabelle actually with the classical way of following this with crystal violet. So she was staining with crystal violet, and then the biofilms were harvested at different times, and you can see how the wild type grows, forms a biofilm, and because the medium is not being replenished, then the biofilm begins to go away. But any of the two mutants, the delta BFD or the BFRB star, they don't form a biofilm. They form an initial biofilm that we detected about 24 hours and no more. So we were curious, and then so what happened here is, in order to do microscopy, is probably best to use a strain that is expressing fluorescent protein from a neutral site in the chromosome. So active bacteria will fluoresce, and then in this case we have a yellow fluorescent protein, and so active cells are, viable cells are yellow, and so here you're looking at the biofilm from the top down, XY axis, and then this will, for example, XZ axis. This is the thickness of the biofilm, and you have yellow fluorescence of these wild type cells, which were harvested at about 60 hours, I believe. So what happens is, in the mutant biofilms, basically most of the cells are dead. So there's a situation apparently where the disruption of iron homeostasis really seems to create a problem with the maintenance of the biofilm. There is some formation of biofilm, but then maintenance of that biofilm seems to be difficult to do. And so we wanted to, and we still want to understand what are the repercussions of creating this iron starvation in the bacterial cells. And so these experiments are with planktonic cells. We have something similar that's being analyzed with biofilms right now, but too early to disclose it in a forum like this. So we grew the biofilms to 36 hours. So when the cells are secreting a lot of pyoverdin, so they are really iron starved, so we know what's going on, we harvest them, and then we did a proteomic analysis. And so the data summarized as follows. So the x-axis is the log2 of the fold change. And the fold change is the abundance ratio of mutant divided by wild type. So here, where is larger than 1, it's going to be, the proteins here are going to be more abundant in the mutant than in the wild type. And here, they're going to be less abundant in the mutant than in the wild type. So we can then this is a summary of how this can be put into context. And we have sulfur metabolism, amino acid metabolism, quorum sensing. Several pathways are affected significantly by the mutation. Okay, and just to give you an idea of how. So here's a bar, a heat map where green is no change. Anything that's bluer to purple is less abundant in the mutant, and anything that's red and brown is more abundant in the mutant. So this is the synthesis of several amino acids whose skeletons are derived from the TCA cycle. A lot of the enzymes are really reduced in the mutant. And we thought, okay, are we going to observe only those iron-containing enzymes being affected? And so we are showing those enzymes that have iron in boxes like this. So not all of them require iron. So probably what's happening is this whole situation starts with less iron being incorporated into into some enzymes. Those enzymes begin to actually affect the cycles where they work, and everything else can be then cascade from there. And so in agreement with that, what we have is that when we analyze amino acid content in the cells, and this was done by NMR, we can get that most amino acids that were in the previous chart are, reduced in concentration in the cells. And interestingly enough, cysteine and glutathione are significantly reduced, indicating potential oxidative stress. In agreement, the proteomics pathway shows that proteins that are involved in red here in importing sulfate, reducing it to sulfide to make cysteine, they are all overabundant in the mutant, indicating that yes, there's sulfur starvation. So there's a potential connection between iron and sulfur that hadn't been seen before. And so these are, just the beginning. We need to understand how. So clearly, then, these results suggest that this target might be interesting to pursue. And so that was that really enticed us to try to think, how is it that we can come up with molecules that bind selectively here in this crevice and prevent the binding of BFD? So that's the next task. Can we come up with such molecules? And so we decided to start. So the title you can't read here, but it is fragment-based drug discovery. Okay, so the idea of fragments is very enticing, especially for academic laboratories. We don't have huge budgets and access to hundreds of thousands of compound libraries, right? So we use informatics, what we know about the structure of the complex, right, and some medicinal chemistry in order to design a 2-fragment library. So fragments are molecules that are 200 Daltons or less, okay, and which are known to bind with high ligand efficiency. So it's different from with large affinity. They are small molecules, so they're not going to bind very tightly, right? But they are going to find those little crevices and then latch onto them and inform us about those little crevices, okay? And so we did, you use saturation transfer NMR, which is ligand-based. So this molecule is 500,000 Daltons, so we're not going to see the signals from the protein. So we're using the ligand, that is the small molecule, right? And then out of that screen of the 200 compounds, we determined that 18 fragments bind BFRB. So now the question is, all those fragments can bind anywhere in BFRB. BFRB is a 500,000 Dalton protein, a very large molecule. How do we know they bind at the binding site, at the BFD binding site? So the solution to that was to take those 18 molecules and screen them again, but this time in the presence of a little bit of BFD. So, of course, the small molecules are not going to compete with BFD effectively, right? If BFD is going to take them out. So if these signals disappear in the presence of BFD, then that means that the fragments bind at the BFD binding site. So of the 18 fragments, we came up with 6 that bind specifically at the BFD binding site. So now these molecules bind with about 1.5 to 3 millimolar binding affinity. So that's not enough to actually do some meaningful in-cell work. So we needed to figure out how to get there. And so the first thing is to do a secondary screen, and then we determine their KDs, and in this case we use surface plasmon resonance to determine that. So this validates the binding. So we have more than one type of binding. You really don't want to spend a lot of time in the future developing things if you're not absolutely sure that it's binding where you think it should bind. And so once we determined that these actually were all nicely done by SPR, so it corroborates binding, then we needed to crystal structure or structural information. And I'll spend a little bit of time just talking about that. So how we did this experiment, basically we can grow crystals of the bacterioferritin, and then we can soak these crystals in a solution that contains the ligand. And crystals of proteins are about 80% water. So these molecules are going to just go through the crystal and they're going to find a place to bind if that's what they want to do. And so we found that this molecule, for example, this talimid molecule binds here. And I'm putting this in here because that's really the leucine 5 tyrosine 2 cleft that I had, the hotspot region that I had identified earlier. So nicely, X-ray crystallography corroborates where this molecule binds. Now the question is, how do we grow these molecules? How do we make them of higher binding affinity, right? And that's why it was worthwhile to be patient and wait for the crystal structure. Because what this crystal structure is telling us, hey, do not modify it from here, right? Do not do chemistry here because you're going to mess up your binding. On the other hand, you can extend here, you can extend here, right? So it already tells you where you should be doing your organic chemistry design. And so we went and decided that after, so this is the organic synthesis is done by Richard Bunce at Oklahoma State University. We've been collaborating ever since I was there. And so we designed the molecules, and Rich comes up with really nice synthetic schemes. And so what we did is grow the fragment to increase potency. And after many, many compounds and many tests and many, this lasted almost 4 years. We decided that this was the best place to extend into this region, into this cleft. And so you make, the best way we found, and I guess most medicinal chemists work the same way, you make small libraries of these derivatives, you test them. And so we wanted to do 2 things simultaneously. One is to do the in vitro KD and see that, you measure a KD and see, that you're improving the binding affinity. But with antibiotics, it's very important that from a very early on in the process, those antibiotics are also tested for activity against the cell. Because actually some retrospectives from pharma have shown how a lot of money and time were invested in getting these exquisitely beautiful binding complexes, only to find out at the end that they did not permeate the gram-negative barrier, okay? So you want to incorporate that at the very beginning. Rules are being gradually developed for permeation of gram-negatives. So if you can, you should incorporate those, potential rules into your design. And so there's a lot to be done here. And then for this molecule, this improved molecule, you can see how we have gone into this cleft here. And this is a crystal structure of one of the earlier derivatives, right? And I can show you here. So this is electron density of the fragment. You can look at hydrogen bonding, waters of crystallization, et cetera. And then this is, so these two rings are joined together by this linker that has one carbon. We decided that also play with the linker, so we have a higher carbon, and the binding affinity goes from about 1.2 millimolar here to about, I think is 20 or 30 micromolar here. I can't remember, I can't see it. And then 0.4 micromolar here. So nice progression of binding affinity. And also the IC50, not active. The IC50 of this molecule is about 80 micromolar, and the IC50 of this molecule against Pseudomonas cells in planktonic culture is 42 micromolar. Okay, so you have increase in binding affinity in vitro, but you should also have increase in, susceptibility of the cells. And this is a figure that this is the cleft. This is how the molecule fits beautifully into this cleft, and now it has a 0.4 micromolar binding affinity, and we are still trying to refine it to make it better. So now we can test these molecules, right? And so this is an interesting slide, I think, in general, because this is remember, we grow these cells that express a yellow fluorescent protein. So the untreated cells, we grow these biofilms to become mature biofilms. So a minimum of 48 hours of growth. Below that, the biofilms are not mature. And so mature biofilms can be treated, for example, with 25 times the MIC or 50 times the MIC of ciprofloxacin, okay? And you can see yellow. They're sitting there smiling at the antibiotic. 50 times its MIC. If you were to use 2 times or 3 times the MIC in planktonic cells, you wouldn't be able to count any cells, right? So that's what I was telling you about the tolerance of biofilm cells. So these cells are tolerant because if we dissolve them into planktonic, then the cipra is going to kill them. Same thing, 25 times or 50 times the MIC of tobramycin. Same situation. One of the few antibiotics that can kill biofilm cells that are mature biofilms is colistin. And colistin is considered as a last resort antibiotic because of its high toxicity. And so at 25 times the MIC, it begins to kill. It kills completely the biofilm at 50 times the MIC. And this is just the percent survival, integrating the fluorescence intensity using COMSTAT. But now let's look at our molecules. So this is the untreated. This is 0.6 times the IC50 of the one carbon in the bridge molecule. 1.2, basically the biofilm is 90% approximately reduced. Then in this case we have the 3 carbons in the bridge, this molecule, and so bottom line, our molecules are bactericidal against bacteria in biofilms. And I emphasize the IC50 here, okay? So what this means is that in planktonic culture, we are not killing the cells. We're merely slowing down their growth. But if we use that, then the concentration, the IC50, the concentration that prevents 50% of the growth in planktonic culture, we can kill the biofilm cells, right? Basically the opposite to what antibiotics are doing. antibiotics kill very well the planktonic cells, and they can't kill the biofilm cells. We have a situation when we have this target that is the opposite. And so now probes, chemical probes can be binding almost anywhere inside the cell. We just want to make sure that our probes are hitting the BFRB-BFD interaction. And so what we did is culture the cells and then treat them with a concentration that would tell us that about 50% of the cells survive, right? And so we got close to that value after counting. And so then we resort to our gel electrophoresis trick again. And so this is the recombinant protein. These are cells, wild-type cells that have not been treated with our compound, and these have been treated with our compound, right? And so what we see is that the band, the iron stored in BFRB, the band is thicker here than it is here. So you can integrate these bands and then you can plot them here, and you can see that in the treated cells, there's almost 3 times as much iron in the bacterioferritin than in the untreated cells, right? So this in itself tells us that the compounds can penetrate the gram-negative envelope. They can engage the bacterioferritin in the cytosol and inhibit mobilization of iron, okay? And then when one accounts for all the irons in the cell, then the treated cells have a lot more iron because all that iron is trapped irreversibly in the bacterioferritin. So basically the same result as we obtained with the mutants, but this time with chemical treatment using the compounds. And finally, I'm going to show you this. So it is, you notice that we have gone to about 10% survival of biofilms and things like that. With colistin, we've gone to the same result. So I believe that treating biofilms is going to be in the future a situation where we're going to have combination therapy. And so we're beginning to think into that, anticipating that. And so what we've done is keep the concentration of the compound the same, okay? And then we have then treated at the same time with 25 times or 50 times, for example, in this case, colistin, right? And you have, if you compare these bars here with the colistin alone or with the combination therapy, there's at least 2 logs of difference, right? And then in the situation of tobramycin with this compound, we get up to 4 logs of extra killing with the combination therapy. Look at the percent survival. This is a logarithmic scale. So now we're beginning to think that actually the combination therapy should be done sequentially. And so we're getting better results than these if we treat first with our compounds and later with the antibiotic. So that is what we're beginning to learn. I don't have that to show yet, but with that, I want to stop, and then I want to, acknowledge the hard work, the talent of people in my group.